राजा। Raja, am I audible?
అన్నా
Am I audible?
కొంచెం వాయిస్ కొంచెం ఆడిబుల్ అన్న మన వాయిస్ క్లియర్ కొంచెం అవును సార్
Okay, okay.
వినిపించట్లేదు. మా మాట వినిపిస్తుందా అన్న?
Yes, 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 yes.
ఓకే సార్ ఆ బ్యాగ్ తీసేసి ఎందుకంటే ఇది ఇబ్బంది అవుతుంది అని ఈ బ్యాగ్ మీరు ఉంటుంది
Hello, can you hear me?
Yes, sir. Yes, sir.
Okay,
Good afternoon, sir. Very good afternoon, sir.
good afternoon.
Good afternoon, sir.
Good afternoon.
Sir, video is only off is coming, sir. Video. Can you make some adjustment for video? Yes, now it is perfect, sir. Perfect, sir.
Hmm.
Yes. Thank you, sir. So just we will start within 2 minutes, sir.
Right.
Yes.
All participants are online or they are in a classroom?
All are in like through few people they are watching through YouTube, sir. So like Zoom account we have limited to only 100, sir. Some of our students they are watching through YouTube, sir.
Okay.
Yeah, Dr. Raja, can you start the session?
Okay. Good afternoon, everyone. This is Dr. Rajaram Mohan Rao, Associate Professor, Department of Pharmacology, Shri Vishnu College of Pharmacy. Welcome back to the fourth day of FDP SGP program. I hope all you had a fruitful and engaging experience from the past 3 days. I'm looking forward to have another insightful day today. So before we begin, allow me to take a moment to introduce our institution. Now I invite Dr. Pawan Kumar Samudala, Head Department of Pharmacology, to introduce our today's speaker, P.V. Bharatan sir.
Yeah, thank you, Dr. Raja. Very good afternoon to everyone. So it is my great pleasure and honor to introduce our distinguished speaker for today's faculty development program session, Professor P.V. Bharatam sir. He is head of the Department of Medicinal Chemistry in Naipur Mohali. Professor Bharatam sir, he is a highly respected academician as well as researcher in the field of medicinal chemistry as well as pharmacoinformatics. Currently, he is serving as a professor in the Department of Medicinal Chemistry at National Institute of Pharmaceutical Education and Research, Mohali. He is having very impressive research experience of over 38 years, including international assignments in the USA and Germany. He has made significant contributions to areas such as theoretical organic chemistry, molecular modeling, organic synthesis, and pharmacoinformatics. His teaching journey spans nearly 3 decades, during which he has guided and mentored numerous students at both master's level as well as the doctoral level. So far, sir has supervised more than 49 PhD students as well as over 225 member of master students. Professor Bharatam sir's prophylactic research output includes more than 358 publications with more than 8,300 citations with an H-index of 47. It reflects his influence in the scientific community. He is a recipient of several prestigious awards such as the Ranbaxy Research Award, OPPI Scientist Award, CSIR Medal, and also he holds fellowships from the Indian Academy of Sciences as well as the prestigious Royal Society of Chemistry, London. In addition to his academic and research roles, he held several key administrative positions at NIPER, including Dean, Associate Dean, and Head of the departments. And also, Sir also served as a institution director for a few months. We are truly privileged to have Professor Bharatam sir with us today. His insights and experience will undoubtedly add immense value to our ongoing journey of faculty development program. So with this brief introduction, I'm requesting Bharatam sir to start his session. Thank you, sir. Thank you so much for accepting our invitation to act as the eminent speaker for faculty development program. Thank you so much, sir.
Namaste, everybody. It is a very pleasant occasion to join the faculty development program. You are all experienced teachers. I just have to share some of my experiences with you. You already are providing excellent training to the students of pharmaceutical sciences across the country. So I will share my slides. Can you see my PowerPoint presentation?
Yes, sir.
Okay. So I have to put it in full screen mode. Okay. Fine. I hope you can see it clearly and you can hear my voice clearly.
Yes, sir.
I'm starting. If any trouble, you tell me.
Okay, sir.
Now, the title of my lecture is about the anti-cancer drug design. Design in the sense not just computer-aided drug design, also drug discovery is also included, design and discovery. Here I am going to talk about the synthesis of compounds as well as the biology of the anti-cancer compounds. So this topic, initially I will focus on the computer-aided drug design. So, but what is the use for you people? In this process, in my lecture, I will tell you the methods of research. This is only an example I am giving, but similar methods can be adopted in any research topic. And for the teaching purpose also, when you are going to teach in your classes, you can probably take the clues from the experience I have in teaching at Naipur Mohali for the past 25 years. I was teaching the topic of pharmacoinformatics. So to begin with, what is pharmacoinformatics? Some of you probably know today what is it. But in the year 2023, when I started this department, in the whole world, nobody was knowing the term pharmacoinformatics. But at that time, Government of India recognized me and invited me to Naipur Mohali. They said that do something big in computer-aided methods. That is where they gave sufficient money also. They gave 1 crore rupees in the year 2023, no, 2003, to start this department called pharmacoinformatics. After that, we got money from DBT, that is another 1.5 crores. And then we have got money from DST, DBT, and then CSIR. Total till now, we have invested about 50 crore rupees on the establishment of facilities in the Department of Pharmacoinformatics. And then we teach the master's MS program in pharmacoinformatics to the students who already completed B Form and cleared an upper entrance test. We offer MS program, MS in pharmacoinformatics to about 15 students every year. And these students are going into industry. Several people are working in Reddy's, Ranbaxy, AstraZeneca, Bristol Myers, and in such companies. And many of them are in teaching also. So right now I will talk about pharmacoinformatics in the next few slides. But rest of my lecture will be based on topoisomerase 2. It is a target for anti-cancer drug discovery. Then I will talk about the synthesis of the compounds and biology of these compounds. So what is the term pharmacoinformatics? It means pharmacon, that is a drug. Informatics, we all know today, I don't have to explain to this generation of teachers and students. Now we all know we are using information technology every day. So the question in the year 2003 was that if biology people are using bioinformatics, chemistry people are teaching chemoinformatics, and physics people are doing, immunology people are doing, many varieties of scientists are working in information technology applied to their field. Then why pharmacy people should not have a topic which is directly related to their teaching and research? That is why we started this department called pharmacoinformatics. In fact, we realized that almost all the bioinformatics scientists in our country, they invariably mention that we use our bioinformatics for drug discovery purpose. If you can use, why cannot we use it? So we are pharmacy people, we are in the field of drug discovery. We do drug formulations, we work in drug toxicity, we work in drug metabolism, we work in drug discovery, as well as clinical trials. So we know better the details of the drug discovery. So we will learn from bioinformatics, chemoinformatics, computer programming, and database management, artificial intelligence from all subjects, and we shall create a department called pharmacoinformatics. This was what was the vision in the year 2003. After 22 years, I can tell that we are very, very successful and we are very happy. So this was supported by Government of India. Whenever we sent a proposal, they happily encouraged us. And that is why we are able to reach this level of satisfaction and happiness. Later, what is the topic? This discipline covers all topics of pharmaceutical sciences and some topics of the computer science, and also a few topics of medicine. Then we have concern which is concerned with molecular modeling and simulation of drug behavior, and subsequently apply that for drug interaction analysis, drug excipient interactions, drug delivery, drug solubility, drug tablet formation, and the various issues of pharmaceutics, as well as drug metabolism, immunology, toxicology, and all other topics. The techniques employed include homology modeling, molecular docking, molecular dynamics, virtual screening, artificial intelligence, etc. Now, this topic is now giving a overall picture, this particular slide. We start our work on one side with from structural bioinformatics, on another side from the 3D chemoinformatics. We bring the data from chemoinformatics and the bioinformatics, and finally we integrate the data and also integrate the tools, and finally come up with the pharmacoinformatics. We always do virtual screening. As a part of that, we have molecular docking, dynamics, and then we have QACR, we have the de novo design, we use chemoinformatics, and many other technologies. For that, we need to learn computer programming. That means any teacher from your batch, if you spend some time to learn computer programming and apply to the pharmaceutical sciences. For example, you know very well, if we know programming, we can apply to telecommunications. We can apply to engineering. We can apply to railway reservation booking. We can apply for Google searching. We can apply for everything and anything in this world. Okay, everything in the world can be handled with the help of information technology. So we should be able to have hold, and we should be able to learn from computer science. For that, the first thing is to learn computer programming. Can we teach computer programming to pharmacy students? First year, second year, third year, any year. Why don't we teach? Oh, sir, it is not there part of the PCI syllabus. Then why we should teach? PCA syllabus, it is not there. We shall go and tell them why it is not there. If the entire world is learning and pharmacy people should not learn, is it? It is a bad practice. So some of you who are associated with PCI, please intimate PCI that we should adopt pharmacoinformatics in our all colleges. We should teach molecular modeling. We should teach artificial intelligence. We should teach computer-aided drug design. We should teach database management. And we should use the machine learning methods. And for that, we need the Department of Pharmacoinformatics. Otherwise, at least we should have a particular course, one course. Don't make it elective. If it is elective, everybody will not learn. Only a few people will learn. Also, if the proper teacher is not available in our college, our principal sir will tell, अरे भाई हमारे कॉलेज में ना वो टॉपिक ठीक से पढ़ाए नहीं पाएंगे हम. Because of that, you take something else. He will guide the students not to take this course. So what I request PCI and also pharmacy college principals is that please introduce one full course, a regular course, not elective course in pharmacoinformatics, because it is very, very relevant today. For that, if you want, I will write the books for that. I have already written many book chapters. I have published more than 375 research articles and book chapters. So I have the experience. I am ready to write a book on this pharmacoinformatics. Only thing I am waiting for my retirement. The moment I retire, I will write the book. So by that time, if PCI will introduce this course at every level, I will suggest you teach it in the B Farm first year itself. Why to go to B Farm third year or fourth year? Because programming is a skill which is required in everything else. A student who can do programming will be successful in his life, in every part of his life. Okay, whether it is pharmaceutics or whether it is pharmacology, whether it is pharmacognosy or any topic associated with pharmaceutical sciences, he can do better job if he can do programming. Programming is such a skill every student should learn, whether it is in the syllabus or not. Okay, so if you have an excellent IT college in your campus, I know I visited your campus many times. When Vishnu College campus has got IT department, why the IT department teacher cannot come and teach extra classes in the pharmacy department for the students of the pharmacy B Farm first year, so that they become meritorious students in the long run. I will tell you, if I am able to stand in front of you and your teacher, Professor Pavan Kumar, told that I have some experience and I have some achievements, it is all because I learned computer programming in the year 1982 in Bhimavaram town, where there was not even a single computer. Whole Bhimavaram town, there was no single computer. But through postal coaching, I learned computer programming from Annamalai University. So that is why I am able to stand in front of you. Otherwise, I am not worth anything. So what I am telling is that teach programming, teach pharmacoinformatics, teach this target identification, target validation, then 3D database management, then virtual screening, hit identification, lead identification, and lead optimization. All these things to your pharmacy students. Do not consider it as an elective course. Consider this as a regular course from today onwards. Don't wait. Don't wait. When somebody from Naipur Mohali will invite me, I will spend 3 months in Naipur Mohali. After that, I will come and teach pharmacoinformatics. Don't do that. That means we are postponing and maybe we never do it. So learn programming from your colleague who is living on your own campus. Maybe you spend 1 hour per week. You will be able to learn. You have a laptop. I know all of you have got laptops. All of you have desktops. Use this for learning. Now I will go to the topic called topoisomerases. What is topoisomerase? It is an enzyme. We all need it. We all need information about the enzymes. Enzyme, I am asking, how do we understand enzyme? We understand enzyme from the biology, friends. We understand from the catalysis that enzyme is doing. We understand the details from the cell biologist. We understand the details from the molecular biologist. We understand the details of the structure of topoisomerase from the structural biologist. And then we can understand the details from the computational biologist. Computational biologist will provide much more details at the atomic level. Which atom is interested in performing hydrogen bonding? Which atom is important for the hydrophobic interactions, van der Waals interactions? Which atom is interested in Coulombic interactions? Like that, we can get many details from the computational details of the enzyme, any enzyme. I will provide the details of topoisomerase in the next few minutes. So, what is this enzyme? What is it doing? It binds to DNA. It binds to DNA, same DNA 2 times. And then it breaks the DNA. Which is very strange, no? there is an enzyme which is created to break the DNA. And then after breaking, it will also reformat. It looks like our Hindu culture. There was a person, probably God. Some people call him God, and he did something. His father told, "Kill your mother." He immediately killed, and he immediately asked father, "Please make my mother alive." This is Lord Parshuram. Okay. Likewise, our topoisomerase, what it is doing, it is breaking the DNA and resealing the DNA, both. Then why does it break? Is it for fun? No. It is to perform isomerization of the topology of the DNA. DNA has got topological problems which are causing its deactivation. DNA becomes inactive if it has got some troubles. I will discuss in the next slide. But to remove those troubles, somebody created this enzyme called topoisomerase, and this will capture the DNA, break the DNA. After releasing the trouble, it will reseal the DNA. That is the purpose of the topoisomerase. So what is this? You can see that topoisomerase 1 and topoisomerase 2. There are two types. Topoisomerase 1 actually causes the topoisomerase 1 causes the where is my pointer? Topoisomerase 1 causes the breaking of the DNA, only one strand of the DNA. Here, only one strand. But topoisomerase 2 causes the breaking of both the strands with the help of the chemistry called phosphorylation. There is a tyrosine molecule. The tyrosine molecule inside the enzyme, OH group of the tyrosine, attacks the DNA and breaks the PO bond and makes a PO bond. As a result, it breaks the DNA at two sides. Topoisomerase 2 breaks on both sides and then pulls the DNA away. Once the DNA pulls it away, a space is created. That space is about the diameter of about 17 angstrom. That is good enough to pass the rest of the DNA, and thus it relaxes the problems are faced by DNA. Okay, so that is the chemistry. So there are varieties of details. I don't want to bother you with those details. Human topoisomerase 2 alpha is responsible for the correcting the DNA. Correcting means making the inactive DNA into active DNA. If active DNA is available, what happens? Cell proliferation happens. If cell proliferation is happening, that leads to the more and more active DNA is available. That means more and more growth of the cells happen. If cells growth is happening rapidly, that is called cancer. So if the topoisomerase is doing its job, that means cancer is growing. So I want that topoisomerase should not do its job. That means I want to find an inhibitor for the topoisomerase. This is the idea. That is where we start looking at the molecules, macromolecules. Let us look at the topoisomerase. What I'm showing is a 3D structure of topoisomerase. Any scientist who want to become successful, he should develop the habit of thinking in 3 dimensions. If you can think in 3 dimensions, you are guaranteed success, 100%. That is what they teach in engineering colleges. What they teach to the civil engineers. Design a house, design a building, design a college, design a town, design Amaravati. Engineers are doing now. Okay. So civil engineers are there to think in 3 dimensions. Computer science engineers are also available to do something called computer-aided design of cars, motorcycles, then the aeroplanes, then everything and anything you want. First, they want to design using computers. Even a simple thing called lock. If you want to design a modern lock, you can do it first on computer and then do it. Old technologies are being modified and updated with the help of computers by thinking in 3 dimensions. Okay, now I want to think about topos in 3 dimensions. So look at the structure. The structure, there are 2 structures on the screen. The left structure on the left-hand side is one. The structure on the left-hand side is the same. Same thing I am showing in 2 directions. One direction is called the where N terminal domain is clearly visible. C terminal is not clearly visible. In the other pose, C terminal is clearly visible. N terminal is not clearly visible. I want you all to look at the protein structure very clearly, very closely. Okay, then we can proceed. I thought someone is asking a question. Is there any question? Okay, if there are no question, I can continue. This thinking in 3D will tell me that you see that pink color. Do you see anything pink color in between?
Yes, sir.
That pink colored portion is actually the DNA. The enzyme, the green colored part is one monomer, and the blue colored, cyan colored is another monomer, and the partly pink colored is the DNA. So this DNA, the topoisomerase holding on to DNA and bend the DNA by 150 degrees, bending the DNA. After that, ATP binds. We all know what is ATP. You know who identified ATP? ATP was identified by Professor Yella Pragada. He is from Bhimavaram. I hope you all know.
Yes, sir.
Bhimavaram scientists found ATP, and same ATP we are trying to understand how it is actually influencing the enzyme action of the topoisomerase. If I understand the cavity in which the ATP is binding, I will know how the ATP is getting hydrolyzed. Hydrolysis of ATP gives rise to inorganic phosphate and ADP. That inorganic phosphate is required in another part of the enzyme catalysis. This is the catalytic cycle of the enzyme. In the beginning, at structure A, B, C, D, E, do you see A, B, C, D, E, F, G? These are the points. Let us consider A. A is the beginning of the enzyme catalysis. In the beginning, enzyme is like this. The 2 monomers. After some time, enzyme comes like this. Can you see my hands? I hope so. Enzyme comes closer, N terminal comes closer. And then it pushes the DNA binds. And then it pushes the DNA down. And then C terminal opens. And then C terminal closes. N terminal opens. C, N, C, N, opening and closing. That means this enzyme, this is a dimer. It can open up and close, but it is not doing that. It is doing this. Okay. If you remember Mahabharata, then there was a Rakshas called Jarasandh. Krishna told Bhim that you break Jarasandha into two parts and throw this part on the right side, the other part on the left side, and then so that it does not form again. Likewise, our topoisomerase is similar to our Jarasandha. It automatically forms this kind of formation. Okay. In the meantime, it will clear and correct the twisted DNA. So this is entire catalytic cycle A, B, C, D, E, F. Once we think in 3 dimensions, the whole thing will become clear. I will go to the next slide. Topoisomerase catalytic inhibitors. Now we want to know what kind of inhibitor we can design. We go back to the structure again. Wherever we want to do anything, we look at the structure. This is very important. Suppose you want to go to Vishnu College. Vishnu College, where is the IT college? Where is the pharmacy college? Where is the girls college? Where is the nursery college? You want to know, you have to think in 3 dimensions. Anything you enter into Vishnu College, you don't know where to go further. What to do? You first come to the gate. There is a security officer. You first talk to him. He will tell you where to go. Similarly, you have to think, you have to come back to structure every time. Every time you have to come back to structure. Okay. It is very similar to the example. Suppose you landed in Hyderabad, you don't know where to go. You don't know anything in Hyderabad. You went in direction, you did not find what you wanted. What to do? First come back to Secunderabad station. Then ask somebody. Somebody will give the answer. Okay. So likewise, you have to start from a particular common point. That common point is the 3D structure of the enzyme. So what is the 3D structure telling? That there is an ATP binding domain. You can design drugs at that point. There is a merberon binding domain. You can design drugs for that. There is an etoposide binding domain. You can design drugs at that point. There is a DNA binding domain. You can design drugs at that point. There is a DNA resealing domain. You can design drugs for that. You can go to something called ICRF domain. You can design drugs for that. The enzyme is going through this process. That means you can design drug at this point or you can design drug at this point. You want to stop this. Okay. This is the aim. If this is continuing, your enzyme is producing the improved DNA, better DNA. You don't want the better DNA. If there is that better DNA, your cancer is growing. So you don't want that. So that is why you should start thinking in the 3D, 3-dimensional structure. So for about the drugs also, you should think in 3D. About the macromolecule also, we should think in 3D. After doing that, we will come across with the 2 pathways. One is called inhibitor pathway. Another is called the poison pathway. Topo2 inhibitors are compounds and Topo2 poisons are another set of compounds. Topo2 inhibitors are on the left-hand side. Topo2 poisons description is on the right-hand side. Both classes of compounds are pretty good for the drug discovery. Now we can take examples of the already known compounds and we can design. Now come back to the structure. Every time, whenever you get into trouble, come back to the beginning. Structure. In our day-to-day life also, we should do this. First, we have to understand the problem. To understand the problem, you have to go to the root cause of the problem. Okay? So, what is the root cause? Okay, suppose you find your young student, you have a young son or daughter at the age of 18 is behaving in a different way compared to what is expected from you and from the society. Then you will have to find out what is the root cause. Is there anybody troubling mentally? Is there anybody giving drugs to your child? Is there anything that your child is not able to understand the chemistry, biology that is being taught? What is the root cause? Then you will be able to solve the problem of your daughter, your son. Similarly, You have to come to the root cause. Root cause is 3D. I'm telling you now, these are the, here you can see the cross. You see the cross that is appearing. The cross is, that is where we can inhibit the enzymatic action. These are the compounds that are already known. Suramine and eclarubicin, they can cause inhibition at that binding pocket. There is one more. Here also I can inhibit. At this pocket also, the same compounds can bind. Suramine and eclarubicin. But in the next step, see, there are compound novobiosine, QAP1. These are the compounds which efficiently bind at the step 3 of the enzymatic action. Now you can also block the step 4. Step 4, you can block with the help of merberon and sodium silicate. These are the two compounds which are useful there. Then if you go to the next one, that is step 5. Here you can, there are so many compounds. These are all called poisons. Topoisomerase poisons. All the other compounds are topoisomerase inhibitors. So these are the compounds which are blocking the resealing process, and thus they are exhibiting the property called uh, the DNA topoisomerase poison property. Now let us go to the next stage. Here at the step 6 also I can cause. Okay, I can cause at this table. This is nothing but compounds called ICRF. This is all known chemistry. What to do? So that is what we did. We have learned all the known chemistry at Naipur Mohali and we published this paper. What are the contents of this paper? I will briefly tell you. First thing we did was started thinking in 3 dimensions. Then we started looking at each and every cavity. Then we looked at the ATP binding domain. If I keep an inhibitor at the ATP binding domain, it is possible to inhibit the entire catalytic cycle. If the catalytic cycle is blocked, cancer is not growing. That is good news for us, everybody. So for that, we have understood the ATP binding domain. Inside the ATP binding domain, what are all the amino acids? There is probably amino acid 342, which is a histidine. Another amino acid 347, which is an aspartic acid. Likewise, we looked at the details and we looked at how magnesium 2 plus is binding in the ATP binding domain, how it is causing the catalysis of the ATP, and then we understood how I can design a new molecules in the same cavity. That novobiosine is doing the job. Now, which compound I can create which is very similar to novobiosine? These are the questions that we asked. And then we solved and published this paper. To do that, what we did? First, we did chemoinformatics. Then what we did QSAR. Then we did virtual screening. After that, Lipinski rule of 5. After that, we employed molecular docking. Then we employed molecular, what is called de novo design. Then we employed molecular dynamics. After that, we employed artificial intelligence. When we employed these many computational methods in the department of pharmacoinformatics, we got these kind of details. What are these details? Magnesium 2 plus is a part of the cavity. Water, 2 water molecules are part of the cavity. Asparagine 91, isoleucine 141, serine 149, tyrosine 165, glycine 164. These are all the amino acids which are contributing to the formation of the cavity. And then our drug molecule. This is the drug molecule which we first docked. This is called imidazopyridine derivative. We performed the docking and then found out where are the hydrogen bonding interactions? Where are the pharmacophore? Where are the hydrophobic interactions? For example, this pyridine ring, it gives you van der Waals forces, that means pi stacking forces, and thus it is hydrophobic in nature. But this carboxyl group is providing hydrogen bonds. That means this is forming uh, hydrogen bonding interactions where between the oxygen and the oxygen of the carboxylic backbone of the tyrosine, of the topoisomerase. Now, likewise, we have done the docking and then we perform the synthesis. We synthesize these compounds. The synthetic scheme I am not showing, but it is not difficult at all. You have to start with 2-amino pyridine. You have to condense with some interesting cyclic compounds, and those cyclic compounds then can be converted into aromatic through the with the help of the aromatization. I can synthesize these compounds, which are known as imidazopyridine derivatives. I can synthesize many of them. This is what we have done. So after that, we performed this study, in vitro study. What are the in vitro studies? Enzyme-based assay have been done. Decatenation assay, relaxation assay, cleavage complex assay, anti-topo2-1 assay. These are the 4 enzymatic assays we have done. Then we have done cell-based assays. Toxicity to cancer cell, is it there or not? Toxicity to normal cell is there or not? Likewise, we have done several cell-based studies. After doing the in silico, after doing the pharmacoinformatics, we were able to do the various various experimental methods. We could design the experimental methods. Which kind of biological assay to be conducted for testing a compound? Which kind of biological analysis should not be done? Those details were generated using molecular modeling. So for that, we have taken the target molecule as 1ZXM. This is called PDB code. This PDB ID, if we download, we can actually utilize that very clearly. And there are so many details how to perform docking. Once you do docking, you will get the result. A few compounds may bind at the ATP binding site, but we cannot afford to do that because any compound which binds to ATP will bind to many other targets. If it is binding to many other targets, how it may be good for binding the ATP of topoisomerase, but it may be bad if I give a compound which is different from ATP binding domain, ATP. So we need to learn more. And then when we docked compounds, it has become clear that some compounds are going into the pink colored regions. You see the cavity, you can see that there is a window through which you can see where our compound is bound. There are a few structures which are plural. It is shown in the yellow color. A few compounds are shown in the pink color. So what is the difference? A few compounds are binding deeply inside the ATP binding domain. Many compounds cannot go to the deep pocket. Okay, what is the meaning of that? Very simple meaning. Some compounds can go to the ATP binding domain and stop the ATP action. But there are several other compounds which cannot go to the deep into the cavity. They are on the outside. So these compounds which are going deeply inside will behave in one way. Those who do not go deeply inside behave in a different way. So that some of them are agonists, other are partial agonists. There are several other compounds which do not bind and do not show action. They are antagonists. So we can define which are the compounds are agonists, which are the compounds antagonists from this kind of analysis. What is shown on the left-hand side It is a 3D structure with the help of a software called Schrodinger software, and then making a choice that show me the secondary structure details, that is alpha helix, beta sheet, and other details. But when you go to the surface level data, there is a region where the surface is negatively charged. There is a region where the surface is positively charged, relatively, on a relative scale. So out of that, then with reference to the binding pocket, we are able to understand the pharmacophoric features of the enzyme. That's what we have done. The result is shown on the right hand side. What is that? Here, this is the surface of the macromolecule when the many molecules are getting docked. The docked molecules are superimposed on one another. That is why you do not see which one is better. But in terms of the whether it is an agonist or partial agonist, to know that this is good enough, but it is not sufficient. We have got to prove it with the help of pharmacology. Initial ideas will come from this docking result. Then we can find the reality after doing the pharmacology comparisons. 60% of the molecular volume is protruding outside from the cavity. Now, this is a table of docking scores. It is like giving marks. We give marks to our students. When we give marks, a particular student may be getting 23%. Another student may be getting 93%. We know the difference. We don't need to know the background of the student. By the time the student is sitting in the classroom, if after taking 5 classes, we are all very clear, this boy is paying attention. That girl is more confident. Another girl is more talkative. She can express the things very well. She will become a teacher. Another scientist, another student will not do any pharmaceutical science. I am clear. After getting the degree, he will do something else. He will do motorcycle business maybe. He will not do pharmacy. You can clearly tell, even though they are in the first semester itself. Very similarly, our docking approach can tell us how much marks we have to give to each and every molecule. That the whole thing is expressed in terms of scores. That is called docking scores. This is a table showing the docking scores of many compounds. Here, look at this glide score. This is a column which is important. Glide score is telling that these are the values. Okay. Now, these are the values. These are the docking values. If we can compare with that of the standard novobiocin or merberon, we will find that the docking scores -5 and everything minus close to 5. If then the lead compound also is close to 5, -5, and thus you can safely declare that these molecules could not, because the scores are so less than that of the PNP, they do not bind in the ATP binding domain, they may bind somewhere else. This is the message. Then we have done the phosphorylation assay. ATP is domain binds to ATP and then gets converted into ADP. For that small chemistry, we have to do the biological experiment and prove that with a change in the time, the biology is behaving before inhibiting with my compound, after inhibiting with my compound. In both cases, we are getting the same kind of a curve. That implies that the drug binding is nothing to do with the binding of the ATP or phosphate binding domain. So that means our strategy is partially okay, but partially not correct. We have to change the strategy. So if not the ATP binding domain, where is the binding site for desired molecules? This question. This question is not easy to answer. This question troubled us for more than 9 months. At that time, we have decided that we have to dock the molecules in merberon binding domain. We have to dock the molecules at the etoposide binding domain. We have to dock the molecules in the GTP binding domain or ATP binding domain, or all of them in one go, and then compare the results. Then after doing that, it has become clear that these compounds that are synthesized, they do not bind to the N-terminal domain. They do not bind in the ATP binding domain. They bind at the merberon binding domain. Merberon is already known to be somewhat lead compound. We can collect some amount of merberon and inhibit, or whether it is showing the antagonistic activity or agonistic activity, we can check. That is all giving that merberon is a good lead compound, but it does not become drug. It never became drug. Why it did not become drug? Because though it is inhibiting, it does not have the proper pharmacokinetics properties. Inhibition is pharmacodynamic property. Pharmacokinetics is also required so that the drug reaches the point of action. So then we wanted to know where is the merberon binding domain. That itself is not known. So probably merberon binding is close to the etoposide binding domain. We can write a very beautiful paper. Somebody may publish it, but it is of no use if I cannot solve the problem. What is the problem? Where are these compounds synthesized or binding? This is the question. For that, merberon was suggested as that type possible. Then we have to dock. Then we have to compare the docking results. Merberon has no effect on either enzyme DNA binding or ATP hydrolysis. That means it is not binding in the ATP binding domain. Merberon does not intercalate with the DNA. That means it cannot bind to DNA and cause the problem. It is not binding at the DNA binding domain, or it is not disturbing the DNA binding. Metformin acts primarily by blocking the 2,2 alpha mediated DNA deviation. So then we came to know that merberon completes the etoposide and reduce the etoposide induced DNA cleavage. So that has given clue. So is it the binding site of merberon, is it close to that of the DNA breaking process? Yes, that's what we found. And then we started docking there. These are some of the results. Docking score is better. Then we docked all our synthesized compounds in the same cavity as that of the ATP binding domain. Then we came with a few compounds are good. They are analogous to ATP. So this is one of the results. This is about the merberon, how merberon interacts with the DNA and how it interacts with the amino acids nearby, and then how it produces the covalent bond and how it breaks the covalent bond. These are some of the questions which appear at the level of atomic level. You will realize that many pharmacologists, many biotechnology people, many pharmacognosy people, many medicinal chemistry people, they don't have time to ask this question. Pharmacoinformatics people have got the time to ask this question. What is the binding site? Where is the hydrogen bond? Where is the docking result? Which one is better than the other? So that kind of decisions were taken. Questions were asked and decisions were taken. And finally, these compounds were suggested and their pK values were obtained. These are all the compounds which are showing better binding affinity than the merberon. So based on that concept, our team has started synthesizing more compounds, more docking, more compounds. And the in vitro docking, results are like this. See the relative kDNA decatenation assay. For compound 5A is pretty good. For compound 6G, 5G, 6A and 6B, it is very small. So from the preliminary investigation, we can compare the results of the computational modeling and the in vitro analysis. After that, we can do another test. So based on the docking score and the decatenation assay, is there any correlation? That we can do. After that, we can draw plots of the in vitro biology versus the experimental biology. In silico biology, we can compare attenuation of the etoposide induced the cleavage complex formation. This is the biological assay, which was successfully carried out, and then we could identify many new compounds. So parent series, there is one series of compounds, there is another series of compounds. First series of compounds were binding at the ATP binding domain. Second series is binding at the merberon binding domain. Both are belonging to the same class of chemical species. Chemistry is same, but biology is different. How do you distinguish this? You can do it by doing the computational biology, molecular modeling, and pharmacoinformatics. When we do that, we come to know that a few compounds are binding at this particular spot, stopping the ATP binding. They are ATP competitive, and they are similar to novobiosine. But another few compounds are binding at this step 4. This is step 4. This is step 5. A few compounds are stopping the step 4 of the catalytic cycle. A few other compounds are stopping the step 5 of the catalytic cycle, but both of them belong to the same chemical class. So these kind of details we obtained. Then we proposed that switch in the site of binding is happening in this particular cavity. Then we published several papers. These are the 3 papers which were published and gave excellent results. And then switch in the site of inhibition. Strategy for structure-based discovery of human topoisomerase 2-alpha catalytic inhibitors. This work was done in the collaboration of Professor S.K. Guchayat, Uttam Banerjee, and other scientists in our institute. Then I thank all of them also. I thank you all for listening carefully to my lecture. And these are the people who have worked in this project. And that means pharmacoinformatics is the beginning and in vivo biology is in the end. In between this, we can do lots of drug discovery. Thank you very much.
Thank you, sir. Yeah, dear participants, if you have any questions.
Hello. Hello.
Yeah, please.
Good afternoon, sir. This is Dr. Purushottam Banjare, Assistant Professor, Centurion University of Technology and Management.
Okay.
Sir, very interesting session, sir. Sir, I have a query that, sir, I have performed the virtual screening with the help of QSAR model, and then I have performed the molecular dynamics for
Okay,
the best compound.
okay.
So do I need to perform molecular dynamics?
See, there are many different varieties of information we get. QSAR provides one kind of
Hello.
information. QSAR
Yes, sir.
provides one kind of information, and molecular dynamics provides
Yes, sir.
completely different information.
Yes,
What
sir.
kind of information is required to judge the quality of our work, to
Yes, sir.
suggest the compound for synthesis, or to perform the biology? What kind of further information is required? Depending on that, we should do the dynamics. For example,
Uh,
dynamics takes
yes, sir.
more time, and
Yes, sir.
we may not have the facility to do dynamics in every college. Then what to do? We do dynamics only when we require the delta E values. How much contribution for the delta E is from the van der Waals forces? How much contribution is from the hydrophobic and hydrophilic forces? If we want to know this difference, and then I want to increase the hydrophobic component in my molecule. If I want to take that decision, first I have to find out, I have to do molecular dynamics and find out whether the hydrophilicity is great or hydrophobicity is great in performing the interactions. If the hydrophobicity is small, I will increase hydrophobicity. Hydrophilicity is small, I will increase hydrophilicity. So likewise, I can take decisions. So when I am ready to take such decisions, it is important to take molecular dynamics. Otherwise, just to show that I have facility, I have done the dynamics, we should not do. We should do only when some science is emerging out of the work that we are going to do.
Sir, actually, mechanistic interpretation of QSAR models and the interpretation of docking both suggested the same type of result, similar type of result. It means the same functionality is responsible for the activity.
Yes, that is good enough. So
Yes, sir.
with molecular dynamics, if you can do, you may do on only 2 or 3 molecules, not on
Okay.
30 molecules. Okay.
Okay. Actually,
And
reviewers are again and again asking the same, sir, for the dynamics.
I know there are several Babcock reviewers who don't understand science, but
Yes, sir.
they only understand technology.
Okay.
They insist. Sometimes I write back to them that you don't know anything.
Okay.
There is no additional science coming from the molecular dynamics. I don't want to do it. I tell them.
Okay. Okay.
I sometimes send a rebuttal, and the editor takes a decision. If he is convinced with the reviewer, he will reject my paper. If he is convinced with me, he accepts the
Yes, sir.
paper. But
Yes,
sometimes
sir.
we do molecular dynamics. We
Okay.
do molecular dynamics on 2, 3 compounds only, and then say that this
Yes,
is
sir.
good enough. You publish the paper.
Yes, sir.
We do that.
Thank you, sir.
Okay.
Yeah. Any other questions? Yeah. Thank you, sir. Once again, I extend my sincere thanks for accepting our invitation, sir. So today talk is very much insightful to all of us. So especially we will try to incorporate coding course to all our pharma first year students, sir, with help of our engineering colleges. So we will try to discuss this thing. Then also we have understood that role of informatics in drug design as well as role of topoisomerase, especially in the cancer research. So thank you, sir. Thank you so much once again.
Okay, very nice, very nice. Giving a lecture in this faculty development program. I hope all the teachers who attended this FDP will go back and teach in a better way and improve the quality of our students. Best wishes.
Definitely, sir. Definitely. Thank you, sir. Thank you so much.
Thank you, sir.
Yeah, dear participants, tomorrow's session will start from 11 a.m. only, not from 2 p.m. Tomorrow's session will start from 11 a.m., not from 2 p.m. So same thing we will keep in group also. Thank you. Thank you very much.
Dear participants, you can find feedback link in the group. Okay, we'll be sharing the feedback link in a few minutes in the group. Thank you.